Welcome to this eukaryotic gene expression basics and benefits. In the last class we discussed about expressing genes in transgenic animals. We starting from transgenic mice and how genes are being expressed in number of other mammals as well, including domestic animals, and we also discussed at length what are the strategies that are used for expressing genes in animal systems and how both for basic research as well as for applied research it has been very useful. Not only in the we have been able to understand the function of genes, the function of promoters by generating transgenic mice and transgenic animal models. We have also been able to express a number of useful genes for making recombinant proteins and recombinant pharmaceuticals, and so both basic as well as benefits arising out of this transgenic technology has been discussed in the last lecture in the animal systems. Today we are going to spend some time to see how people are trying to express genes in plants, how people are trying to generate transgenic plants, and what are the basic technology that is involved in expressing genes in plants, and how the society got benefited by the generation of transgenic plants. I also would like to caution here that this is a subject that is highly controversial. There has been a lot of controversy on the introduction of transgenic plants into the environment. There are a number of groups which oppose to the introduction of transgenic plants into the fields and the use of transgenic animals both for agriculture as well as for medical purposes. But what I am trying to focus here mainly in this lecture is not to get into the controversies too much, but try to tell you the science of it and also try to project some of the positive aspects, namely how expressing some of the useful genes in plants led to the development of very novel traits, and how society actually got benefited by introducing these transgenic plants in agriculture. So just as we have discussed about introducing the genes in transgenic plants, how people use something like microarray injection into the fertilized egg, or you can introduce genes into the embryonic stem cells using viral vectors, and how you can carry out germline transfer into animals to generate transgenic mice. In the case of plants also, people use about two basic technologies for introducing genes into plants. One is the Agrobacterium mediated gene transfer, which can be called as the biological method of gene delivery, and in cases where the Agrobacterium mediated gene transfer is not very efficient, people use what is called as a biolistics. Just as we have seen in the case of gene delivery, in case of gene therapy, you can actually use gene gun or biolistics to pump or to forcefully deliver genes coated on gold particles into the plant cells. So there are also several other minor techniques that people use for generating transgenic plants, but to the paucity of time we will confine ourselves to discussing these two major methods of generating transgenic plants, how transgenic plants can be generated by delivering genes through the Agrobacterium mediated gene transfer or by using gene gun. So the basic strategy has been somatic tissues of plants grown in culture can be transformed in the laboratory with the desired gene using one of the above methods and grown into mature plants with flowers. So following gene transfer, the transgene is incorporated into the pollen and eggs and therefore is passed on to the next generation. So this is basically germline gene transfer, just as we have seen in the case of transgenic animals, where we are introducing genes into the fertilized egg onto the yeast cell, so that not only the animal which is generated, but the successive generation also carries the transgene. The same way, by introducing genes through the Agrobacterium mediated gene delivery or by bialistic methods, these transgenes also gets incorporated into the pollen and eggs, and therefore the transgene gets passed on from generation to generation. So what is this Agrobacterium mediated gene transfer? So just as animal viruses are used for gene delivery vehicles for the introduction and expression of genes in animal cells, bacteria which infect plants can be exploited for delivering genes into plant cells. And one of the most commonly used bacteria for the generation of transgenic plants is a bacterium called Agrobacterium tumefaciens, which is a plant pathogen normally found in the soil. So it is a soil microbe. Agrobacterium tumefaciens causes a disease called as crown gall disease. It is actually shown here. This is the crown gall in a variety of dicotyledon plants, especially members of the rose family such as apple, pear, peach, cherry, almond, raspberry, and roses, and so on. So it is basically a plant pathogen. Plants infected with this bacterium develop large tumor-like swellings called as galls. One of them is shown here. That typically occur at the crown of the plant just above the soil level. So, following infection, the bacterium transfers part of its DNA into the plant, and this DNA then integrates into the plant's genome, causing the production of tumors and associated changes in the plant mesobolome. So, here is a bacterium that actually naturally infects the plants, and during the infection process it actually delivers its DNA into the plant and therefore expresses its genes and also alter the plant metabolism. So, just as in the case of the when we discuss gene therapy, how animal viruses to also do the same thing, they infect the animal cells, push the genome into the animal cells and then exploit the host machinery for its own survival. In the same way, here is a plant pathogen, a bacterium, which also infects the plants, pushes its DNA, which goes and integrates the plant genome and then directs the plants to produce certain metabolites which is useful to you for the propagation of the bacterium. So, let us now see how this basic phenomenon has been used for generating a vector system for delivering genes into the plants. So, the process of infection, as I just described, the Agrobacterium tumefaciens infects plants through wounds. The motile bacterial cells are attracted to the wound site by certain phenolic compounds such as acetylsalicylate, which is actually secreted by the plant at the wound site, and these compounds are sensed by specific chemotactic receptors which are present on the bacterial membrane. So, the bacteria are attracted to some of the chemoattractants which are actually secreted by the wound. The wound may be happen naturally or during the propagation. So, this when a plant gets wounded, it secretes certain chemical compounds, one of them is called acetylsalicylate, and the receptors present on the bacterial surface sense this and the bacteria now get attracted towards the wound. So, this is basically called as chemotaxis. In addition, these compounds which are secreted by the plant wounds such as acetylsalicylate at higher concentrations activate certain genes known as virulence genes which is present in the bacteria, and these genes are encoded by a bacterial plasmid known as the Ti plasmid. Ti stands for tumor inducing plasmid. And as a result of the expression of these genes encoded by the bacterial plasmid, certain proteins known as permeases are actually inserted into the bacterial cell membrane for the uptake of compounds that are actually produced by the tumors. And acetylsalicylate also causes the production of an endonuclease that excises the part of the Ti plasmid turned as tDNA and transfers into the plant genome. So, let us now discuss in detail how exactly this whole process of infection by the Agrobacterium takes place using a cartoon. So, here are the soil bacteria. So, when the plant gets wounded, these wounds secrete certain compounds such as the acetylsalicylate, and by chemotaxis, the receptor present on the bacterial cell membrane, the bacteria senses these compounds and gets attracted towards the plant wound, and once it gets attracted towards the plant wound, the bacterium attaches to the plant cell and the bacterium, this is the main bacterial chromosome, and here you have a bacterial plasmid, and a part of this bacterial DNA called as tDNA now leaves the bacterium and enters the plant wound through specific bridges that are actually formed between the plant cell and the bacterial cell, and then this tDNA gets integrated into the main bacterial chromosome, and as a result of the expression of some of these virulent genes, the plant is supposed to have called or gets transformed, just as some of the animal viruses when they go inside and cause transformation leading to cancer. Here also, because of the introduction of this tDNA and expression of the plasmid coded genes, the plant cell starts producing the Vir genes of the bacterium now starts producing certain factors which act as growth factors for the plants and promotes rapid cell division. And as a result, the plant cells around the transformed region develop very rapidly, and you get what is called a tumor or the crown gall. So, this is the basic physiology or genetics by which the bacterium induces tumor formation in the plants called as the crown gall. So, let us now focus on this plasmid which is carried by this Agrobacterium tumefaciens known as the Ti plasmid or tumor inducing plasmid, because that is what is very essential for generating transgenic plants. Now, the Ti plasmid is nothing but a circular bacterial plasmid of about 200 kilobases in size, and this is known as the tumor inducing plasmid. It is responsible for the production of tumor as well as for transformation. Now, the Ti plasmid transfers and inserts a region of the plasmid known as the tDNA into the host chromosome. So, here you have the Agrobacterium tumefaciens. It has its main bacterial chromosome. It also has this Ti plasmid or the tumor inducing plasmid, and within this plasmid there is a small region denoted here in the pink color known as the tDNA, and it is this tDNA that gets entered into the plant genome. The tDNA actually codes for production of tumor as well as synthesis of certain molecules known as opines. Now these opines are the ones that promote growth of the plant cells. They act as plant hormones and promote the growth and multiplication of the plant cells, leading to the formation of the tumor. Now this TI plasmid is very important, because if the TI plasmid is lost, as for example when you grow the bacterium above 28 degrees centigrade, this plasmid is lost, and if you now take such bacteria which lack the plasmids, they cannot induce these crown galls or tumors, and therefore they become avirulent, clearly indicating that for induction of the tumor or the for the transformation, this TI plasmid is very essential. Now there are a number of TI plasmids depending upon what kind of opines they produce. There are what are called as nopalin plasmids, which actually carry gene for synthesizing a compound called as nopalin in the plant and for the utilization in the bacteria, and tumors that arise out of these nopalins differentiate into shooty masses called as teratomas. Okay. There are other kind of plasmids called as octopine plasmids. They carry genes which synthesize compounds known as octopines in the plant and on the catabolism of the bacteria, and in this, when bacteria carrying these octopine plasmids infect plants, the tumors do not differentiate but remain as callus tissue. So depending upon what kind of opines are produced, the morphology of the tumor varies. There are what are called as certain Agrobacterium tumefacients carry what are called as agropine plasmids. They carry genes for agropine synthesis and catabolism, and here also the tumors do not differentiate and they die out. And finally there are what are called as RI plasmids. They induce hairy root disease on some plants and the crown gall in others. They have agropine type genes and may have segments from both nopalin and octopine plasmids. So basically try to tell you there are different types of TI plasmids. Some of them produce nopalin, some of them produce octopines, agropines, and so on and so forth. Depending upon what kind of these opines are produced, the kind of crown gall that is produced or the tumor that is produced varies. Now, so this is the cartoon of a TI plasmid. How does the TI plasmid look? Basically the TI plasmid has what is called as a left border and a right border, and the genes that actually code for the auxins, opines and cytokines. It is these genes that actually are responsible for promoting the growth and differentiation of plant cells, leading to the formation of the tumor. For the insertion of this T region into the main bacterial chromosome, you require this left border and right border. Any region that is put behind the left and right border will now get inserted into the plant genome, leading to the integration of this region into the plant genome. So the strategy of making a transgenic plant has been to take these genes that actually codes for some of these auxins, cytokines and opines, and in their place put your gene of your interest. So the gene of your interest is inserted between the right border and the left border, so that these genes are replaced by your gene of your interest. In addition, you also include an origin of replication of E. coli origin of replication, so that once you make the recombinant construct, there can be made in large amounts in E. coli, just as we have done for many of the other mammalian and yeast vectors and so on and so forth. So the strategy is to introduce an origin of replication to the TI plasmid, so that this plasmid can be propagated in E. coli cells. Then clone the gene of interest into the TI plasmid by replacing the genes in the tDNA with the gene of your interest between the right and left border, and then introduce this engineered TI plasmid back into the Agrobacterium and allow it to infect the plant. So what happens when this tDNA TI plasmid gets inside the plant cell, the region between the right and left border now gets integrated into the plant genome, and therefore your gene of your interest with its promoter now gets expressed. So this is the basic strategy by which Agrobacterium mediated tumefacients transgenesis is carried out in plants. So basically you construct what are called as binary vectors. First you generate a TI plasmid with your gene of interest, that is in the place of the vir genes, you replace the vir genes or the virulence genes with your gene of your interest, and since you require the virulence genes for the transformation, you actually now express the vir genes on another plasmid called the helper vector. So you have two plasmids, you transform both these plasmids into the bacterium and then make this bacterium carrying these recombinant plasmids, make it infect into a plant, and you generate a transgenic plant. So the basic procedure of creation of a transgenic plant using an Agrobacterium tumefaciens is the plasmids, that is the disarmed TI plasmid with the gene of interest as well as the helper vector containing the vir genes, are transfected into Agrobacterium tumefaciens. The plant cell culture is then infected with these Agrobacterium tumefaciens carrying both these plasmids, and the products of which genes excise the gene of interest with tDNA and transfer it to the plant genome, and plant cells which carry these plasmids are now selected by incubating them in the presence of kanamycin, growing them in the presence of kanamycin. So, all those cells which are not taken up the DNA, which have not been infected with the Agrobacterium tumefaciens will die. So, only those which have got infected will now survive, and then you confirm the transgene by PCR, just as we have done in the case of the transgenic mice. It is a detailed protocol of how the first binary vector was actually constructed and was published in Nature 1983 is given in this particular paper. A binary plant vector strategy based on separation of virulent T region in Agrobacterium tumefaciens stay plasmid. One can go and read this paper in detail to understand how exactly these binary vectors were constructed and how they were used for generating transgenic plants. Now, the Agrobacterium mediated tumefaciens is primarily infects the dicotyledon plants. It cannot really infect monocot plants. So, during the early time of transgenesis of plants, the Agrobacterium mediated gene transfer was primarily used for generating transgenic dicot plants. But since this bacterium cannot infect monocot plants, an alternate strategy was developed for introducing genes into monocotyledon plants, and that is the gene gun or the biolistic method or the particle bombardment method. Again, this we have discussed in the case of gene therapy, wherein actually again you use a gene gun, where you take your DNA of your interest, mix it with gold particles and shoot them into the tissue using gene gun. In the same way, you use a gene gun based system for delivering gold coated DNA, and you just using a helium gas, when you push this gold coated particles coated with DNA with high pressure using helium gas, and the tissue culture cells or the plant callus cells now get transfected by these gold particles carrying the DNA, and they get this is how the transformation is carried out. So, in the case of monocots, at least during the early years, the generation of transgenic plants was carried out using the biolistic method or the gene gun method using gold particles coated with DNA of your interest, because the Agrobacterium tumefaciens did not infect monocots. But then later in 1994 to 1997, Agrobacterium mediated transfer protocols were also developed for monocot species, especially for food crops like rice, maize and wheat. So, nowadays even monocot plants can be transformed using Agrobacterium mediated tumefaciens. I will not go into the details of it, I have just given the references here for the lack of time. So, one can actually read some of these references to see how these Agrobacterium mediated gene transfer techniques were actually developed for monocots as well. So, this is a paper, efficient transformation of rice mediated by Agrobacterium and sequence analysis of boundaries of the tDNA, published in Plant Journal in 1994, gives how the Agrobacterium mediated transfer was developed for rice. Similarly, in 1996, similar protocol was developed for introducing genes into maize by Agrobacterium mediated tumefaciens, published in Nature Biotechnology, and again in 1997 a similar protocol was developed for introducing genes into wheat, published in Plant Physiology in 1997. So, a number of crop plants can now be gene transfer can be carried out using Agrobacterium mediated gene transfer. Now, what kind of promoters are used for expressing genes in transgenic plants? Just as we have seen in the case of the animal systems, a number of plant promoters are available. This could be viral promoters which infect plants, or it could be natural plant promoters. They could be tissue specific or development stage specific and so on and so forth. But one of the most widely used or most exploited promoters in generating transgenic plants has been derived from a virus called as cauliflower mosaic virus, and this promoter is from a 35S gene. So, this cauliflower mosaic virus 35S promoter is one of the most widely used promoters for expressing genes in generating transgenic plants. So, the 35S promoter is a very strong promoter that is active almost in almost all dicot plants, and it is one of the most extensively used promoters for the transgenic plants. In fact, this is a patent that is owned by Monsanto, and they had a virtual monopoly for using this promoter for generating transgenic plants for a long time. and the patent only expired sometime in 2005. Therefore, anybody can actually use this 35S promoter for generating transgenic plants, both for basic as well as for certain applications. Now, what kind of selectable markers are used for generating transgenic plants, because it is very important. When you do either the gene gun mediated gene transfer or use the Agrobacterium mediated gene transfer, not all cells will get transformed. So, you need to select those cells which have either taken up which got infected with the bacterium or which have taken up the DNA, and therefore, usually the vector that you have used usually has to contain a selectable marker. Some of the common markers which are actually used in the case of transgenic plants has been what is called as a neomycin phosphotransferase gene. There are two such genes called as neomycin phosphotransferase 1 and phosphotransferase 2, NPT1 and NPT2 genes. This was originally isolated from a transposon present in the E. coli bacterium, and the gene actually codes for aminoglycoside 3-phosphotransferase denoted as NPT2, and this enzyme inactivates by phosphorylation a number of aminoglycoside antibiotics such as kanamycin, neomycin, geniticin and paramomycin. So, cells which have not taken up this plasmid containing the selectable marker, they will get killed by these antibiotics, whereas cells which have taken up the antibiotic, because they express this phosphotransferase gene, it will phosphorylate this antibiotic and makes it inactive, therefore, they survive. So, these are you can select those cells which have taken up your gene of your interest, whereas those cells which are not taken up will get killed by the antibiotic. The other very commonly used selection marker in generating transgenic plants has been hygromycin phosphotransferase gene, again derived from Escherichia coli. This gene again codes for hygromycin phosphotransferase. Again, as in the case of the NPT, it actually detoxifies an aminocyclitol antibiotic known as hygromycin. So, basically the enzymes produced by these resistant genes inactivate the antibiotic, and therefore, cells carrying these genes become resistant to the antibiotic, whereas those cells which are not taken up this antibiotic get killed. There is also a non-antibiotic marker gene which is routinely has now been used. Actually, this came up only in 2004, where you can actually use an enzyme called phosphomannose isomerase as a selectable marker gene instead of an antibiotic resistance gene, and this gene, which is called the PMI gene, actually converts mannose 6 phosphate to fructose 6 phosphate, which is a usable carbohydrate. So, if we now have cells which are expressing PMI and which cells which are not expressing MI, if we now use mannose 6 phosphate as a carbon source, in the case of those cells which are expressing the PMI gene, mannose 6 phosphate will get converted to fructose 6 phosphate, which can then go into the glycolysis and the plant can use this carbohydrate. Whereas those which do not express the enzyme, they cannot make use of the mannose 6 phosphate and therefore they die. So, instead of the antibiotic resistance marker, a metabolic enzyme can also be used as a selection marker in the case of generating transgenic plants. So, you have either the NPT1, NPT2, HPT or the PMI genes, and these are the three genes which are routinely used for generation of transgenic plants as selectable markers. Now, what kind of reporter genes are used for generation of transgenic plants? Because suppose I want to study what kind of promoter, what is the nature of the promoter, which cells it is expressed, what are the cis-acting elements of a natural promoter of a gene. If I have to identify, just as promoters have been characterized in the case of transgenic mice, and a very valuable information was obtained on the developmental regulation or tissue specific expression of number of promoters, people have also used this transgenic technology to understand the function or mapping the cis-acting elements of various promoters using the transgenic plants. So, in such cases, the strategy is you take the promoter region of your interest, link it to a reporter gene, introduce them into the plants either by bialistic method or using the Agrobacterium mediated gene transfer, and then see if by activating the reporter gene and studying the reporter gene expression, you can understand what is the cis-acting elements which are present in the promoter region. And one of the most commonly used reporter genes is what is called as the GUS, that is beta glucuronidase. Remember, in the case of the animal cells and in bacteria, we normally use what is called as a beta galactosidase. The E. coli beta galactosidase is normally used for as a reporter in a number of microbial as well as the animal systems. In the case of plant systems, the big glucuronidase or abbreviated as GUS is the most popular reporter gene that is actually used. Now, Gus is a hydrolase that cleaves a wide variety of beta-glucuronides and is most widely used report system for plants. It is very easy to quantify, highly sensitive and very specific, very important characteristics for a very successful reporter gene. And substrates for Gus are available commercially, and you can assay the activity of the reporter gene or the Gus by spectrophotometry, fluorimetry or histochemical methods. So, it is a very convenient and easy assay for detecting the reporter gene expression, and this is the compound, colorless compound that is actually used for the Gus activity measurement. And when this compound, the bromochloroindole beta glucuronide, commonly known as the X glue, when it is normally colorless, but when the Gus gene is expressed, it gets induces oxidative dimerization, leading to the formation of a blue precipitate. So, the appearance of blue color indicates that the expression of the reporter gene. Now, so what I have explained so far is how one can generate transgenic plants. Basically, there are two major methodologies. One is using the Agrobacterium mediated Agrobacterium tumefaciens mediated gene transfer using the tumor inducing plasmid, where you introduce your gene of interest inside the right and left border of the TA plasmid, and then you, by expressing which genes on another plasmid using a binary vector system, you can actually generate a Agrobacterium tumefaciens expressing the transformed these plasmids, and then you make this bacterium infect the plants and then select those cells that are taken up or infected cells by using the appropriate selection markers. And we also discussed one of the most popular promoters that are used for generating transgenic plants is the 30-phase promoter of the coliform mosaic virus. There are also number of other promoters which are used. We will not get into the details. And if you want to assess the activity of a promoter in a plant system, normally reporter genes such as glucuronidase is the Gus is normally used, but people also use luciferase or green fluorescent protein as the reporter gene for assaying plant promoters and generating transgenic mice. Plus, Gus has been the most popular reporter gene that is used for assaying promoter activity in transgenic plants. So, these are some of the basic aspects I have discussed. Now, a number of variations of the some of the features I have discussed are possible, but we will not get into the details or variations of these various protocols, but I have given you basically a general or a basic idea of how do you generate a transgenic plant. And if you want to express a gene of your interest, normally use a very potent constitutive promoter like a 35S promoter, or if you want to assay for a promoter activity of a unknown promoter, you can take the promoter, put it under a reporter gene such as Gus or GFP or luciferase, and then transform the plant and then look for the reporter gene activity in various tissues of the plants. So, having explained the basic aspects of generating transgenic plants, let us now try to spend some time and see what are the benefits that came out of generation of the transgenic plants. Now, one of the basic basic research has got enormously benefited. As I said, people are using transgenic plants for understanding the function of a number of genes. People are also using transgenic plants for understanding the function of a number of promoters. This is the basic aspects of generating transgenic plants. So, by using transgenic plants, one has been able to map a number of tissue specific promoter, developmental stage specific promoters and so on and so forth, and also has been able to study and understand the function of many novel genes. So, it has been extremely used. Transgenic plants has been extremely useful in basic research for understanding gene functions as well as for understanding the promoter activity. In addition, transgenic plants has been extremely useful and has a number of benefits have come out of transgenic plants. So, I am going to now list some of the benefits that came out of generation of transgenic plants. It is not possible for me to explain all the benefits that came out of transgenic plants. So, I have actually made a choice. As examples, I am going to discuss how by generating transgenic plants you can improve nutritional quality of plants, you can develop plants which are resistant to insecticides, you can develop plants which are resistant to certain diseases, you can also develop plants which are resistant to herbicides, you can develop plants which can tolerate salt, high concentration of salt. So, you can grow these plants in marshy areas where normal plants cannot grow. You can actually, by expressing certain genes, especially those encoding antisense RNA, you can delay fruit ripening, which again has a lot of commercial applications. You can also use transgenic plants for the production of a number of biopharmaceuticals, recombinant proteins as well as vaccines. So, let us spend some time to see how by using these two basic approaches, that is either introducing genes using bialistic method or agrobacterium mediated tumefacients, what kind of genes have been introduced in the plants, transgenic plants, what kind of transgenic plants were generated, and how the society got benefited from some of these transgenic plants. Now, one of the most exciting results that came out of generation of transgenic plants, it is what is called popularly known as what is called as the golden rice. Now, we all use the rice. When you take the paddy and then try to put them in rice mills, the husk is actually removed during this milling process. Along with the husk, the beta carotene, the aileron layer which actually contains the beta carotene, is also lost. Therefore, the rice, the polished rice that many of us actually eat, is actually depleted of beta carotene. Now, beta carotene is very important for vision and for the production of vitamin A, which is very essential, and without vitamin A, a number of physiological functions are affected. As you all know, vitamin A actually gets converted to retinoic acid, and retinoic acid is a very important morphogen and is required for development, and also if you consume excess amount of retinoic acid, it can lead to teratogenesis. Similarly, from the vitamin A you get retinal, which is a very important component for vision. So, if you do not take sufficient amounts of vitamin A, it results in vitamin A deficiency, causes blindness and a number of other health problems. So, especially people living in Africa and certain developing countries, because of use of this continuous use of polished rice, and they do not supplement vitamin A by other means such as vegetables and so on and so forth, they virtually develop blindness and number of other health hazards. So, people thought, why can't we develop rice which actually now contains beta carotene. So, what actually was done is, beta carotene is the precursor for vitamin A, and consuming milled rice leads to vitamin A deficiency, especially in countries such as Southeast Asia. Therefore, people ask the question, can we now express the genes that is essential for the synthesis of beta carotene, so that now you have rice which actually contains lots of beta carotene. So, even when you polish this rice, it still has a lot of beta carotene. Now, to make beta carotene in the rice, you have to express the genes which are involved in the biosynthesis of beta carotene, and unfortunately this synthesis of beta carotene is a multi-step process. So, people realize that you need to express more than one gene if you have to make beta carotene in plants. So, in January 2000, a group of European researchers reported that they have succeeded in introducing 3 transgenes into rice that enable these plants to manufacture beta carotene in their endosperm. So, even if you lose the aileron layer and lost the beta carotene, the endosperm still has lots of beta carotene, and therefore such the vitamin A deficiency can be avoided by eating such rice. Here I have just shown you the normal rice, which you can see is white, not it is colorless because it does not contain any carotenoid. Here is a transgenic rice which expresses now about 101.6 milligram per gram of beta carotene, and you can see it is now light yellow, and if you express higher amounts of carotenoids in this rice, you can see it is dark yellow. So, this is one of the golden moments of transgenic research, where people develop transgenic rice containing high amounts of beta carotene, and thus you can enhance the nutritive value of food crops by expressing certain transgenes, and this is one such example. So, this generation of golden rice created huge amount of excitement among researchers and as well as public. Similarly, here are tomatoes which are purple. Why? 2 genes were expressed from snapdragon which induce the production of compounds called as anthocyanins, and when you express these 2 genes leading to the synthesis of anthocyanins, now the tomatoes turn purple. Now, it is not just the color that matters. These anthocyanins offer protection against certain cancers, cardiovascular diseases, age related degenerative diseases, and therefore there is evidence that anthocyanins also have anti-inflammatory activity, promote visual activity, and hinder obesity and diabetes. So, by consuming these kinds of transgenic tomatoes which are actually expressing these anthocyanins, it can actually promote your health. So, again enhancing the nutritive value. Here is another example, genetically stable expression of functional miraculin, a new type of alternative sweetener in transgenic tomato plants. Now, what is this? Now, miraculin is a taste modifying protein actually present in miracle fruit, which are actually the red berries of Richardella delphica, which is a shrub native to West Africa. So, they again took the gene that they again took the gene encoding this miraculin protein and then introduced into tomato plants or other plants. And what happens when you consume these fruits which are expressing this miraculin, the taste changes. So, sour foods such as lemons, limes and grapefruit taste sweet when tasted together with this protein. So, somehow when you express this protein in some of these fruits, the sour taste is converted to sweet taste. So, you can see again enhancing the quality of the food by expressing certain transgenes in plants. Here is another example of benefits of generating transgenic plants by expressing the appropriate genes in plants. Tearless onion. We all know that when you cut the onions, tears roll down ears. Why is that? As onions are sliced, the cells are broken, alinases break down, and sulfoxides are generated. They are converted to sulfinic acids, which are highly unstable, and they rearrange into a volatile gas which diffuse by air and then reaches the eye, and these acids now react with water to form a dilute solution of sulfuric acid, and tear glands produce tears to dilute and flush out this irritant. This is how whenever you cut onions you get tears. Now what happens? You now generate a transgenic anion by expressing a gene that actually downregulates or represses the synthesis of lacrimatory factor synthase. Now this is the protein enzyme that is responsible for the production of this irritant, and if you now use what is called an RNAi technology, I have still not discussed this RNA technology in this course. We are going to discuss this maybe in the next couple of classes. So, if you now repress the expression by using RNAi or RNA interference technology of a synthesis of a enzyme called lacrimatory factor synthase, the conversion of these valuable sulfur compounds into the tearing agent is inhibited. So, now you have onions which does not induce tears. So, you can see by expressing appropriate genes, either by expressing certain enzymes or by inhibiting certain enzymes, expression of certain enzymes, you can have very beneficial effects by generating such kind of a transgenic plants. Here is another. Now this is an example I want to give. We should not be under impression that all these useful traits can be generated only by transgenic plants. Here is another very interesting example, what are called as rainbow cauliflowers, where you have purple, yellow cauliflowers. These were actually produced by traditional breeding methods by a company called Syngenta. Actually it even has a subsidiary in India, right, in Pune, but these were developed by non-transgenic methods. And what is the significance of these? The orange cauliflower has higher than normal levels of beta carotene that encourages healthy skin, just as you have the golden rice with high levels of beta carotene. Here you have a cauliflower containing high levels of beta carotene. In the case of the purple color comes from anthocyanin, again which helps prevent heart disease by slowing blood clotting. So, but the point I want to make here is that these kind of rainbow cauliflowers expressing again having useful traits were actually generated by non-transgenic means. So, let us not be under the impression that interesting traits can be incorporated into some of these agricultural plants not only by transgenic technology, but even by conventional plant breeding. Very useful traits can be introduced into the plants. So, I have given a number of examples to tell you how the nutritive value of food products can be enhanced by generating various transgenic plants by introducing appropriate genes, either by expressing certain genes or by inhibiting the expression of certain genes. Now, the other important example I would like to give is that how you can generate transgenic plants which are resistant to insect pests. Bacillus thuringiensis, for example, is a bacterium that is pathogenic for a number of insect pests. Why is that? Because this bacterium contains a protein called as Bt toxin, Bacillus thuringiensis toxin. So, when insects consume this bacteria produce this toxin, and this toxin kills the insects. So, what do you do now? You now take this Bt gene or the Bt toxin gene and generate a transgenic plant expressing the Bt toxin. So, when the insects now eat the plant leaves or the plant tissue, the Bt toxin now enters the insect gut and it kills the insects. So, you do not have to spray insecticides. So, the use of insecticides can be completely avoided by generating transgenic plants or economically important crop plants expressing toxin gene. They can now develop resistance to major insect pests, that thereby obviating the need for using insecticides. So, you can see by introducing genes like Bt toxin gene into important crop plants and generating these transgenic varieties, they can be made resistant insect resistance. So, you do not have to spray insecticides to kill to prevent insects from destroying these crop plants. Again, disease resistance, Genes that provide resistance against plant viruses have been successfully introduced in a number of crop plants such as tobacco, tomatoes and potatoes. A number of plant viruses cause a wide variety of diseases, especially in many of the fruits like tobacco, tomatoes, potatoes and so on and so forth. Like in tobacco, for example, you have the tobacco mosaic virus and so on and so forth. So whenever these plants get infected with these viruses, they also express certain disease resistant genes, and by engineering these genes which provide disease resistance, you can actually develop these plants which can now resist these viral viruses. They become resistant to many of these viral diseases. Herbicide resistance, again genes for resistance to certain herbicides have been introduced into crop plants so that they can thrive even when exposed to herbicides that kill weeds. So one of the major problems in agriculture is that along with the crop plants or the plant that you want to grow, a number of weeds also grow alongside the plants. So how do we destroy these weeds? Because many of the fertilizers that you use are consumed not only by your crop plant, by your plant of interest, but also by the weedicide, so which is a waste of money. Therefore you want to actually kill the weeds selectively, but not your crop plants. So by engineering herbicide resistance genes into your crop plant of your interest, if you now spray these herbicides, your plant will not get affected, but the weeds which are actually growing along these plants, they get affected by these herbicides. Therefore you can kill all the weeds by using this kind of a herbicide resistant plants. Now the importance about I have told you that you can use, I have given a list of what kind of transgenic plants have been generated. You can use it for nutritional value, herbicide resistance, insect resistance, so on and so forth, but almost more than 99% of the transgenic crops which have commercial value are either herbicide resistance or insect resistant crops. So introducing genes that confer herbicide resistance or insecticide resistance has been one of the major aspects of generating transgenic plants, especially from the agriculture. So the other traits, like various other traits that I have mentioned so far, contributes less than 1% of the total transgenic crops we have today. So majority of the transgenic crops that have been produced today are primarily devoted for developing resistance to certain herbicides or developing resistance to insect pests. So some of these other traits that people are trying to develop in transgenic plants is development of salt tolerance. For example, a large fraction of world's irrigated land cannot be used to grow most important crops because of the saline nature of the soil. The soil is highly saline, so normal plants cannot grow there. But many of the plants actually express what is called a saline resistant genes. When you expose them to many plants which grow near the marshy areas, they are tolerant to the salinity conditions because they express certain genes which confer resistance to salinity. Now if you now take such genes which confer resistance to high salt concentration, put them in your crop plants, you can now grow crop plants in marshy areas which are otherwise not suitable for agriculture. So research have created transgenic tomatoes that grew very well in saline soils. The transgene introduced was actually a sodium proton antipode pump that sequestered excess sodium in the vacuole of the leaf cells. So by expressing a gene that actually codes for a sodium proton antipode pump, the salt that is ingested into the plant is actually sequestered into a vacuole, and therefore the plant is able to survive in high salt conditions. Again, transgenic plants expressing antisense RNA. Now when you express an antisense RNA, the antisense RNA will go and hybridize the sense RNA which is normally present in the cells, and as a result a double strand RNA is formed and translation is blocked. So the RNA will not be translated and protein of your interest will not be produced. So tomatoes, can you now use this kind of antisense by technology to delay ripening of certain fruits? Very famous example has been what is called the flavored sour tomato produced by a company called Calgene. Now most tomatoes that have to be shipped to the market are harvested before they are ripe. Otherwise the ethylene that is synthesized by tomato causes them to ripen and spoil before they reach the customer. So from the place of production, that is from the farm, to carry these genes towards to the actual marketing place, which is usually a town or a city, you have to, if you allow the fruits to ripen on the plant and then transport, most of the tomatoes will get damaged during the transport itself. So there is a tremendous loss. So this is because the tomatoes produce ethylene which is which induces fruit ripening. Now transgenic tomatoes have been constructed that express an antisense RNA complementary to the mRNA for an enzyme involved in ethylene production. Therefore production of ethylene is delayed. Now, these transgenic tomatoes make only 10% of the normal amount of the enzyme, therefore, it delays ethylene production. So, you can pluck them early and then transport slowly, because it is producing much less ethylene, the fruit ripening is delayed, and as a result the damage due to fruit ripening before it reaches the destination can be prevented. So, a company called Calgene actually developed this kind of a tomato and called it as flavor sour tomatoes, but because of a number of opposition for the introduction of some of these transgenic plants or transgenic fruits or edible crops, transgenic frost, there is a lot of resistance for these things. Many of these transgenic fruits never saw the light of the day. Production of therapeutic proteins, just as we have seen that farm animals are being used for production of a number of therapeutic proteins such as factor 8, factor 9 and so growth hormones, so on and so forth. You can also use plants using again transgenic technology for producing a number of recombinant proteins which have of medical or biological importance. I will not go into the details of the production of therapeutic proteins. A number of such recombinant proteins has been produced. It is more or less the same proteins which we discussed in the last class using transgenic animals or using expression system, other expression systems. So, I have just given one review article here. One can go through this review article and see what kind of recombinant proteins have been produced in transgenic plants and does it have any commercial value. But rather I would like to stick myself for the limited amount of time we have to see what kind of vaccines can be produced by using transgenic plants and transgenic technology. Now, as you all know, vaccines are very important. In the two classes before, we had actually discussed about DNA vaccines, how a failed gene therapy technique was actually used for developing a DNA vaccine, and by simply injecting DNA into in a encoded by eukaryotic expression vector, mammalian expression vector, you can actually express the antigenic proteins in the mammalian systems or inside our human or animal tissues, and therefore it will get presented to an MHC class 1 or class 2 system, and you get either a cell mediated immune response or a humoral immune response. So, here the question we are asking is, instead of expressing the gene coding for this antigenic protein either in yeast or E. coli or in the mammalian cells, why don't you now express the gene using a plant promoter, so that now the plants actually produce the viral antigen. Now, this viral or the foreign antigen produced in plants can either be purified, or if you put it in express them in edible parts, by simply eating those edible parts of this plant you can get an immune response, and therefore you can get protected against that particular pathogen. This idea of using plants as vaccine production factories was proposed first by a scientist called Charles Aronson in the Arizona State University, and it is the first paper actually that came out in 1992 in PNAS entitled Expression of Hepatitis B surface antigen in transgenic plants. So, in this paper they actually shown that tobacco plants were genetically transformed with a gene coding for hepatitis B surface antigen linked to a nominally constitutive promoter, and the recombinant hepatitis B surface antigen which was purified from these transgenic tobacco plants had properties very similar to that derived from the serum. No, the traditional way of making hepatitis B vaccine is to purify the virus like particles from the infected human patients, and they found that the hepatitis B surface antigen which is expressed in these transgenic plants is very similar to that produced from the human plasma. And therefore, they concluded that transgenic plants can hold tremendous promise as low cost vaccine production systems. So, instead of taking your gene, putting them in a yeast or putting them in E. coli, and then making this recombinant protein, purifying this yeast and growing them in huge fermenters and then purifying them and then giving it as a vaccine, why can't we now express these genes right in the plants and then see whether I can use plants as vaccine production platforms. Now, in 1995, this is the paper that came out where they actually expressed the rabies virus glycoprotein in transgenic tomatoes, and actually shown that this recombinant protein produced in tomatoes can actually induce an immune response in animal models. Now, in 1996, again Aronson's group actually expressed what is called a Norwalk virus capsid protein in transgenic tobacco as well as potato, and it demonstrated if mice are fed with these potatoes which consume this antigen, potato is expressing antigen, they can develop an immune response. The first human clinical trial of a vaccine antigen produced in plants was done published in 1998. immunogenicity in humans of a recombinant bacterial antigen derived from transgenic potato. So, in this paper they discussed about how mice, when they feed these potatoes which express this hepatitis B surface antigen, they can actually develop antibodies against the hepatitis B surface antigen, or humans which actually consume this raw potatoes expressing the hepatitis B antigen, you can actually demonstrate the presence of antibodies against this viral antigen in their serum, indicating that the antigen which is consumed through the oral route along with these potatoes can actually stimulate our immune system to produce antibodies against the viral antigen. So, this was one of the landmark papers in the area of transgenic plants for use as edible vaccines. So, it generated a lot of excitement, as you do not have to produce these antigens in organisms like yeast or E. coli and then use needles to inject these antigens. By simply consuming fruits or edible portions of a plant expressing the plant antigen, you can get immunized. So, oral vaccines or edible vaccines became a huge generated a lot of excitement among researchers as well as public. So, here is for example, in 2005 they actually demonstrated that the human's hepatitis B surface antigen, which is actually consumed through the oral route, can elicit similar kind of response as generated by the normal hepatitis B antigen injected through the systemic route. So, the human volunteers who consumed these hepatitis B antigen expressing potatoes can develop antibodies in their serum, whereas the control group did not show any antibody titers. But this excitement died down because people soon realized that this concept of edible vaccine also sounds very exciting, cannot really be a commercial success. A nice article actually appeared in Nature Medicine clearly saying that edible vaccines are not ready for the main course. Now, plant-based vaccines face the biggest scientific and regulatory hurdles, because you just cannot express your vaccine antigen in a thing like potato or tomatoes and then simply ask people to eat these tomatoes or potatoes and get immunized, because these are all the dose, the amount of antigen I have to take, the quality, everything has to be properly regulated. Now, one can ask, for example, should I use a big potato or a small potato? Should I use a big banana or a small banana? How do you regulate the dose of antigen that you have consumed? Now, what is the amount of antigen that is produced from different batches of the plants? So, all these things have to be argued out, and so the initial excitement that Charles Arendsen's work actually developed slowly died down, because even Arendsen now said that his original idea of distributing vaccine bearing fruits was naive, because regulatory agencies will not approve vaccines with variable dosings, because you have no control over the expression of the antigens in these plants. It may vary from batch to batch and you have no control over how much you ingest. So, vaccine manufacturers have little reasons to replace the existing production lines, as most vaccines are economically unattractive. So, if vaccines are ultimately presented together with food, the gut's immune system also faces a conundrum. The gut is designed not to react to antigens in the food, but must produce the useful response against vaccine. So, instead of being immunized, patients could even end up being develop tolerance, meaning an immune response against future invaders would be weakened and not intensified. So, this concept of using these edible vaccines slowly died down. But what now Charles Arendsen, who is the pioneer in this technology and now believes, is that instead of the original idea of consuming these fruits as edible vaccines, he now thinks that you can actually take these edible portions of these plants, freeze dry them, and then take this freeze dried material, put it in the form of a capsules, and now you can actually determine how much antigen it actually consume. You can now regulate what dosage it is. So, the concept of edible vaccine has now become what is called a oral vaccine. That is, you make these antigens in plants and then freeze dry them. So, here you can see the freeze dried potato media or freeze dried tomatoes, and then push them into form of capsules, and if you now orally take these capsules, you can develop an immune response. So, the concept of edible vaccine has now slowly changed to oral vaccine, but still, as I speak today, still no company has really come forward to successfully commercialize this concept of plant based vaccines as yet, but it is likely to happen in the future. I think there is a very nice article written by Charles Arendsen. One can download this PDF file from the website called the Upstream and Downstream Manufacturing of Vaccines and Tobacco. This is a facility that is present in called as a Biodesign Institute in the Arizona State University. Primarily, and there is also a nice article which appeared just recently this year on transgenic plant based oral vaccines. One can read this to get excited more about very useful information about using transgenic plants for vaccine manufacture. There is also a lot of controversies on transgenic plants. As I said, introduction of transgenic plants into agriculture has been vigorously opposed by many groups. There are a number of issues that worry the opponents. One of them is the potential risk of transgenes in commercial crops endangering native or non-target species. For example, a gene for herbicide resistance, that is in corn, may escape into a wheat species, and now the wheat may now express the herbicide resistant gene, and therefore it may become much more difficult to control the wheat. Similarly, the gene coding for a Bt toxin expressed in the pollen might endanger pollinators like honeybees. Now, you do not want to kill honeybees because they are natural pollinators, but because they are also insects, they may also die because they consume this Bt toxin producing, they get contaminated by the pollen containing the Bt toxin. So, there are a number of such opinions expressed. But a large number of field studies on transgenic cotton and maize indicated that the reduction in number of certain non-target insects is much smaller than that observed in the fields treated with the insecticides, because the insecticides also pose the same problem. So, there are a number of other things, but the take home message is that despite all these concerns, in United States, for example, more than 80% of the corn, soybeans and cotton grown are all using genetically modified plants, which all have resistance to herbicides or resistant to insecticides. So, a number of transgenic plants have been approved by regulatory agencies both for edible as well as non-edible purposes. The acreage of transgenic crops has gone from virtually 0 in the year 1995 to almost about 345 million acres in 2009. And here are some of the statistics here. If you can go to this website, you can get the things. You can see here from 1996 when the first transgenic crop was introduced till last year, you can see if you look at the total global area of biotech crops has been continuously increasing, both for industrialized countries, developing countries, as well as the total almost 345 million acres is now under cultivation of transgenic crops. Similarly, a number of crops you can see soybean, maize, cotton, canola, there is a steady increase in the growing transgenic crops all over the globe. Similarly, the global area if you go by the traits, the herbicide resistance is the topmost transgenic crops expressing which are resistant to herbicides occupy the top of the chart. This is followed by those which are insecticide resistance or herbicide resistance. So, these are the majority of the transgenic crops that are being produced now, either are herbicide resistant genes, insecticide resistant genes, or and so on and so forth. And you can see the global area of biotech crops from again 1996 to 2006. A record of 14 million farmers in 25 countries planted 135 million hectares in 2009, a sustained increase of 7% on 9 million hectares over 2008. What I am trying to say is that every year the transgenic crop growth is increasing. I think one can just go through some of these statistics to really show that the global status of commercialized crops in 2009. I will not go into the details. There are what are called as terminator genes. Again, there are some concerns. One can go through this and try to understand what these concerns are. I have just listed some of the references here. This reference actually summarizes all the eukaryotic expression systems that we have discussed so far, yeast, insect, plant expression systems. One can go through and understand more. Again, this website actually gives the global status of commercialized biotech or GM crops, and these are some of the biotech companies: Monsanto, BASF, Dow, Bayer, DuPont, Syngenta, some of the leading biotech companies which are actually expressing a number of genes and developing transgenic plants for a number of commercial applications. Thank you.